And make sure you all pick up your study hints before you leave today. I really didn't get to finish going through them. It takes like half an hour to grade one set, so just really haven't had time. But those that I did grade, I put some points on there. The main thing you all should do is after you define the study hints, whatever I'm asking, come up with an example, because that'll help you remember. And I'll give you all an example of that a little bit later. But that's the main thing I saw missing from the study hints. Okay, so one of the study hints requires that you know the organic molecules of life, and those are
oxygen, carbon.
molecules of life, carbohydrates.
Nucleic acid.
Great. Okay. And why are they called molecules of life? They're found in all living things, particularly what component of all living things? Cells, right. So all living things are composed of cells, and those cells are made up of these organic molecules. Last time we kind of went over how these organic molecules also contain energy that can be used to what? Recharge ATP. So carbohydrates are the first of the organic molecules of life that we'll discuss. Carbohydrates are also referred to as sugars and starches, and all carbohydrates contain carbon, hydrogen, and oxygen, just arranged differently. The three classes of carbohydrates are monosaccharides, disaccharides, and polysaccharides. And obviously, the mono, di, and poly, you know what those mean, those prefixes. Mono means one, di, two, poly, many or multiple. Good. So the building blocks of carbs, these are the types of questions I'll be asking on the test for you to recall what the building blocks of each of the molecules of life are. So the building blocks of carbohydrates are monosaccharides. Is everyone clear what I mean when I say building blocks?
They make them up.
Yeah, so they make up the monomers, right. So you'll hear them referred to as monomers or building blocks or subunits. Okay, so they're the component, the small component that makes up the larger molecule. Okay? So the monosaccharides, and in your study hints, hopefully you're using that second page and studying that, because you'll see those on the test. The monosaccharides that you should be aware of are glucose, fructose, and galactose, which are 6 carbon molecules. So glucose contains 6 carbons, we'll show you in a moment. Fructose and galactose also have 6 carbons, but they are all monomers, monosaccharides, also called simple sugars. And you also have ribose and deoxyribose. And you all have heard those before. So remember, ribose and deoxyribose are also monosaccharides, but they contain 5 carbons. And so the monosaccharides again are what? Glucose, galactose, and fructose. Good. And the disaccharides are maltose, sucrose, and lactose. So if you have a disaccharide, it's made up of what? 2 monosaccharides. And you can see those here. So notice that all of the disaccharides up here contain 1 glucose at least, and then the other monosaccharides that we mentioned. So sucrose is made of fructose plus glucose. Maltose is made of two glucose molecules bound together, and those are bound together by not hydrogen, but covalent bonds. And then you have galactose and glucose, which bond to form lactose. So those are the three disaccharides you should be familiar with. The polysaccharides are starch, glycogen, and cellulose. And again, you should know a little bit about these. So starch is the storage form of excess glucose in plants. So I think we may have mentioned last time that when we consume carbohydrates, our digestive system breaks them down, right, to their what? Monomers. Okay, when you all get to the digestive system in 202, you'll go back over these molecules of life because they involve most of the foods that we eat contain those molecules of life. And the reason we consume them, carbs, proteins, lipids, is because of what? To get the energy from them. Okay? And again, in order to get the energy, in fact, what needs the energy? Okay, great. So you all remember. So keep that in mind. So it's the cells that need the energy. So somehow those nutrients have to get from the digestive tract to the cells of my fingertip. Wow, how does that happen? Right, there you go. So that's what the blood is for, to transport the nutrients from the stomach, from the intestines, to the cells of the body. Okay? And in order for those nutrients to get into the blood in the first place, they have to be converted to monomers. That's kind of the basic understanding you should have at this point. So starch has to be broken down to glucose, which is a monosaccharide. Okay? Glycogen, you'll find that glycogen is the way our body stores excess glucose. So again, if you eat 6 Krispy Kreme donuts, you don't need all that glucose that is going into your bloodstream to stay there. So some of the glucose is packed together and stored as glycogen. Okay? So as much as possible, try to visualize this so it makes more sense to you. So glycogen is the storage form of excess glucose in animals, whereas plants store excess glucose in the form of starch. And then cellulose is found in the cell walls of plants, and we consume vegetables mainly for that reason, because of the roughage that it provides to the diet. So cellulose, the interesting thing about cellulose is it's what type of molecule? What type of molecule is cellulose, starch, and glycogen? Cellulose, starch, and glycogen are all polysaccharides. Okay? So in order to get the nutrients from them, they have to be broken down to monosaccharides. The thing about cellulose is our digestive system cannot do that. Okay? So cellulose cannot be broken down by our digestive system. But it's still good for us. In fact, very important for us.
Is that where we get cellulose from?
Completely different thing.
I wasn't sure.
Okay, so so think about that for a moment. So something that we can't even digest is important in our diet. Any idea how that could be? It's probably one of the most important things we can consume. Fiber, right? So cellulose provides fiber. What's so special about that? Okay, so if it cannot be digested, what happens to it? So think about that. So most of the foods we eat are broken down and sent to the bloodstream. Cellulose is not, but it's good for us. So if it's not going into the bloodstream, where is it going? All the way through the digestive tract, right? Fiber pushes everything out. Okay? And you'll see that that's important because buildup of materials in your digestive system, we have bacteria that start acting on those, and they produce toxins, and we need to get that out, especially things like red meat just sitting in your colon. There's evidence that they can produce cancer. So fiber, roughage is good because it pushes all that stuff out so it doesn't sit in the walls.
I want to ask you that. Some fibers aren't they good for healthy use as well? I mean,
Well, yeah, I mean, there are some benefits. The cellulose is just a part of it, but you also have other nutrients within those as well.
okay, I wasn't sure if it was connected to the cellulose or not. I just knew that.
Yes, sir. All right. So polysaccharides. So we can move on through this and get to the lipids. So lipids are what we refer to sometimes as fats. And lipids also contain carbon, hydrogen, and oxygen. And sometimes P, which is what? Phosphorus. Good. So lipids are insoluble in water. What does that mean? So they don't dissolve in water. So you try to mix water and lipids, it just doesn't work. Okay? The main types are triglycerides, which are referred to as neutral fats, phospholipids, steroids, and eicosanoids. And for now, basically just know that those are the types of lipids. So if I give you a list, I might say which of the following is not a lipid or something. Okay, as far as any more details about each of them, no, don't worry about it. Now, phospholipids, we'll talk about in the next chapter. You do need to know that phospholipids are found where? Right, they make up the cell membrane. Good. And fatty acids are the building blocks, one of the building blocks of lipids. And fatty acids look like that molecule there at the bottom. So notice all the carbons there. So that's what makes it what? Organic, all the carbons and hydrogens. So this is one example of a fatty acid, palmitic acid. And you'll know that fatty acids are also referred to as either saturated or unsaturated. And what that has to do with is whether or not there are hydrogen bonds, maximum number of hydrogens bound to carbon. And I don't have a picture of unsaturated, so I'm going to give you all an example of, so you'll know the difference between saturated and unsaturated. Notice all of these carbons are connected to each other by one bond. Okay? These carbons are connected by one bond between each. But this one has two bonds, two covalent bonds. It's called a double bond. So anytime a fatty acid is considered saturated, that means it looks like this, no double bonds. Unsaturated contains carbons and double bonds. Okay? And it's going to look a little different. The reason it's called saturated versus not saturated is because the unsaturated do not have a maximum number of hydrogens. Okay? Okay, so notice if you have a double bond here, you don't have hydrogens up there. So it's not saturated, in other words, with hydrogens is all that means. So the saturation, the word saturation is really referring to the carbon atoms are saturated with a maximum number of hydrogens. If you have the double bonds there, you lose two hydrogen atoms. Okay, so saturated versus unsaturated. And then polyunsaturated would mean what? You may have a double bond here and another double bond there. So you have more than one set of double bonds.
You go back over it.
Over what?
You go forward when you got friends.
For polyunsaturated?
Yes, ma'am.
Yeah, so a polyunsaturated would be one that has, so it may be a double bond here, but there may be another set of double bonds here. Poly meaning many, right? Many double bonds. Okay? All right.
Anytime you say poly,
Right. Poly means many or multiple. Triglycerides or neutral fats, so three. So if you hear a triglyceride, tri meaning three, that means three fatty acids covalently bonded to three carbon alcohol such as and glycerol molecules, and I'll show you a picture in a moment. But each bond formed by dehydration synthesis, you all should be familiar with that. Dehydration synthesis is that reaction that causes what? Monomers to combine to form polymers. And what about hydrolysis? Right, so hydrolysis is the reaction that breaks down polymers to their monomers. And you have that with carbs, lipids, proteins, and nucleic acids. Okay? So on the test, the question may be, which reaction would convert amino acids to a protein? Okay? You get it? Which reaction would convert starch to monosaccharides? Okay, so that's how you have to think about it. Okay, so triglycerides at room temperature are called oils when they are liquid. Triglycerides at room temperature are called fats when they're solid. Okay, so those lipids that are solid at room temperature are called fats. If they're liquid at room temperature, they're called oils. Okay, and mostly when you think of fats, saturated fats come from animals. So that when you eat different types of foods, some lipids are better than others, right? Vegetable oil, for example, is better than lard, because lard comes from animals, animal fat, which is saturated.
Animal fat.
Yes. Okay. And so the primary function of lipids is energy storage, insulation, and shock absorption. And it's found primarily in adipose tissue, fatty tissue. And adipose tissue lies under the skin. Okay, phospholipids. We'll move on from there. But remember that phospholipids have the phosphate group, and the head is hydrophilic. What does that word mean?
Water.
Yeah, water friendly.
Right, water friendly or water loving. And hydrophobic means water fearing, does not like water. So water interacts better with which part? The head. Good. Steroids and cholesterol are also lipids, but they don't have the chain of carbons like the fatty acids. They are more of a ring structure. A steroid is a lipid with 17 units of carbon atoms and 4 rings, so they have ring shapes instead of a long chain. Cholesterol is the parent steroid from which other steroids are synthesized. Cortisol, so you've heard of these hormones, cortisol, progesterone, estrogen, testosterone, all of these are steroid hormones. Okay, then also bile acids involve cholesterol. So remember that cholesterol is a lipid. a type of lipid. Cholesterol synthesized only by animals, especially the liver cells. 15% comes from the diet, 85% is produced by the body. And we'll see in the next chapter that cholesterol is also embedded in the cell membrane along with the phospholipids. And be familiar with the types of cholesterol, good and bad cholesterol. You've heard of HDL and LDL. Which one is good for you? HDL, so high density lipoprotein. Break that word down, lipoprotein. What do you think that means? So the lipo part means lipid, and obviously the second part is protein. So a lipoprotein is a protein that has lipid as a component of it. Okay? So HDL, again, is good cholesterol because it has a lower ratio of lipid to protein. So even though both of them are made of proteins with lipid, HDL has a lower amount of lipid compared to that of protein. And HDL may help to prevent cardiovascular disease. LDL is the bad cholesterol. It has a high ratio of lipids to protein and contributes to cardiovascular disease, the buildup of plaque. And this is obviously going to affect some people more than others. So some people, for example, you have heart disease that runs in your family, then you want to make sure you're not consuming too many foods that are going to have cholesterol and certain types of saturated fats, because they can build up in your arteries, the arteries of your heart, and prevent blood from getting to the muscle cells of your heart.
How do people, do they just take when they have a lot of LDL, and they give you blood pressure medicine or whatever, and they're supposed to eat Cheerios? How does the Cheerios reduce that? Does that have a high amount of HDL
Well,
in it?
I'm not sure how, but it's probably has more to do with the role of, because Cheerios has some fiber in it, so I'm thinking that has something to do with it. I know fiber counteracts bad cholesterol in some respects, so it may help to push it on through. Okay, so that's what steroids look like. So notice again the difference in the structure. It's not a long chain of carbons, but it has those 4 ring structures. Eicosanoids, again, don't worry about these for now. Just know that they are lipids. We'll talk more about their specific functions later on. Proteins are polymers of amino acids. There are 20 different types of amino acids. And the bond is actually covalent, but in proteins the bonds are called peptide bonds. So peptide bonds are found between amino acids within the protein molecule. So amino acids are the building blocks of proteins. And just some terminology here. So if you hear the word peptide, that refers to any molecule composed of 2 or more amino acids. So you can have dipeptide, tripeptide, oligopeptides, polypeptides. Proteins, if it's called a protein, it has more than 50 amino acids, sometimes up to hundreds of amino acids. Okay, so how do you form a protein from amino acids? What type of reaction? What kind of synthesis?
Dehydration.
Good. Dehydration synthesis. A wasp, yes. As long as it stays back there with you all, I'm fine. Okay, so dehydration synthesis, this is a picture of it. Make sure you all understand the difference between dehydration synthesis and hydrolysis. Which one involves water coming in? Okay, so hydrolysis is water coming in to break the bonds between the monomers. Hydrolysis and then dehydration. Dehydration means what? Taking water away. So you're pulling water out in order for the bonds to form between the two monomers. And there's the polypeptide chain. Protein denaturation. One of the things about proteins is that the shape of the protein sometimes determine how the protein works. So if the protein changes shape, it no longer functions the way it's supposed to. So there are factors to consider, such as pH and temperature. So if a protein undergoes a rapid change in pH or temperature, then that can denature the protein and prevent it from working. Sometimes this is reversible, so if you, for example, if you expose the protein to a high pH and then bring it back down to normal, the protein starts working again. But sometimes, if it's the denaturation is severe, or if there's an extreme change, the change is irreversible. Okay, and so the proteins may not be able to go back to working the way they were supposed to. One example of that is cooking an egg. So, what's the best part of an egg, the white or the yolk?
White.
What's so good about the white? So the egg white is made of protein. What protein? Anybody know? You heard of it? Starts with an A? Albumin?
Never heard of that.
Okay, so albumin is a protein that we also have in our blood. And ALBUMIN, albumin. So when you cook an egg, notice that the egg white is clear before you cook it. And after you cook it, it turns what? White. Okay, so what has happened is that protein has denatured. And notice you can't make it go back to being clear again. So that's an example of denaturation that is irreversible. And that's because of the extreme temperature change. Okay. Let's speed this up before the wasp goes crazy. But it's staying up high. It's not even coming down. So just ignore it. Okay. So enzymes. There are a few things you all need to know about enzymes. First of all, enzymes are proteins. Enzymes are proteins. That's number one. And their function is to speed up chemical reactions. There you go. So that's what I'm looking for when I ask you for the function of enzymes.
Can we just say catalyst?
Not if you don't know what catalyst means.
I do. We learned that in like 8th grade.
Okay.
9th grade biology.
Yeah, you can say that, but I may ask it a different way. So if someone memorizes catalyst and doesn't know what that means.
I know what it means.
Yeah, but you should know what that means. So catalyst speeds up things, reactions.
Emphasize speed up, protein speed up what?
Enzymes are proteins that speed up reactions. They permit reactions to occur rapidly at normal body temperature. And we're going to talk about a whole lot of enzymes coming up, especially in 202. One type of enzyme you may be familiar with, lactase. You ever heard of lactase?
Is that not?
Lactase breaks down lactose. What is lactose?
Milk acids.
Lactose? Okay, what about it? So what is it? What type of molecule? What'd you say? Lactose is a disaccharide. And in the digestive tract, it has to be broken down to monosaccharides. So lactase is the enzyme that breaks down lactose to those two monosaccharides. Okay? We also have enzymes that break down starch. So enzymes are really integral to how our body cells work. And so enzymes are necessary. And what this says is it just speeds up a reaction. So you may think, well, they're not that necessary if we don't have them. So what? The reaction just takes longer, right? But some of those reactions may take 100 years. So if it takes 100 years to break down starch to glucose, you're not going to get the benefit of the nutrients from glucose. Okay? So that's just an example of why enzymes are important in being able to speed up reactions. Okay? And you also know that enzymes interact with substrates. I won't go into detail about this. You all should remember some of this from Biology 103. And notice that enzymes, the names of enzymes, usually, not always, but usually end in ASE, named for the substrate in which they have the effect on, such as amylose is going to digest the starch, but it's going to be digested by amylase, which is an enzyme. So again, know that all enzymes are proteins. Okay, this picture just kind of shows you how enzymes work. Again, all they do is speed up a reaction. They don't interfere with any changes of the molecules except for binding them or breaking them apart. Okay, but notice that the enzyme never changes. So the enzyme just connects to those substrates, binds them together, and then the enzyme goes back to doing what it was doing before, just not being changed much at all. Nucleic acids are DNA and RNA. These are the largest molecules in the body. And DNA is located where? Right, in our cell's nucleus, the nuclei of our cells. So nucleic acids, also contain carbon and hydrogen, but also include oxygen, nitrogen, and phosphorus. The building blocks of nucleic acids are nucleotides. Good. And a nucleotide, you should know what a nucleotide is composed of. Nitrogenous base, which are, can you name them? All right, good. So those are the nitrogenous bases. All nucleic acids also have a pentose sugar, which are pentose meaning 5 carbon. We mentioned 2 of those monosaccharides that have 5 carbons. Remember what they were? So what are the sugars that are in DNA? Ribose or the other one? What does the D in DNA stand for? Deoxyribonucleic acid, right? Definitely you all should know what DNA stands for. Deoxyribonucleic acid, and RNA stands for ribonucleic acid. So the ribo is for ribose sugar. The deoxyribose is for the sugar, the pentose sugar, deoxyribose nucleic acid. And then you have the phosphate group. So you all just told me the 4 bases. And one thing about DNA, so you all should have an idea, just in general, the differences between DNA and RNA. What's the difference in the bases? So RNA would have, instead of thymine, would have uracil. Very good. DNA is double-stranded, a double-stranded helix, whereas ribonucleic acid only has 1 strand.
And it's what helps the DNA separate from the virus.
Somewhat. We'll get into that in chapter 3. Okay, so DNA provides instruction for protein synthesis. We'll get into that next week. All right. So that's what the DNA strand looks like. So again, most of this we'll talk about in chapter 3, so don't spend too much time on it here, just the basics. And then ATP So that's your ATP molecule. What does ATP stand for? Okay, you all should know that as well. It's one of those things I can stop you in the hall and ask you, what does ATP stand for? You should be able to tell me. And if you all go to the cafeteria here? Okay, every now and then I'll come to your table and ask, what is ATP? What is RNA? Or I'll find you wherever you are, Walmart, McDonald's. Okay. So that's it. Yes. Yes.